Hey there, curious minds! Ignite your learning journey with us. A very warm welcome to my channel. Are you ready to embark on an extraordinary learning adventure? So, I'm getting very excited. Please forgive me if there are any mistakes. This is my first video. So, we all believe that education is the key to unlocking your true potential and achieving your dream. So why delay? Let's get started. Here is your first question, and each question carries 5 marks. The answer of phylum Nematoda is pseudocoelom because pseudocoelom is a form of coelom that isn't made up of mesoderm like true coelomates, and it is located between mesodermal and endodermal tissue. Next, class average is pneumatic bones because it makes the skeleton light in weight, which makes it easy to fly, and pneumatic bones are hollow bones filled with air. The answer of superclass Achnatha is mouth without jaw. Next, phylum Porifera is a non-motile animal because non-motile animals are those organisms that are not capable of moving from one place to another. The answer of class Osteichthyes is fin rays. Fin rays help fishes to control body position and to generate force during locomotion. The second question is multiple alleles act not at individual level but only at population level. Justify the statement with the example of inheritance of ABO blood group in human. So I hope you all know what is multiple allele. So here we have to first write the definition of multiple allele. When a particular gene has 3 or more alleles or alternative variants, it is called multiple allele. Multiple allelism is observed in a population and not in an individual. An individual is diploid, that means only 2 alleles of a gene be present in an individual. And the reason is human gene I for isohemagglutinin, exist 3 allelic forms IA, IB and IO. IA and IB produce type A and type B antigens respectively on the cell surface of RBCs, whereas IO or I is a recessive allele and doesn't produce any antigen. So for intents, an offspring that get both IA and IB alleles will have blood type AB, and that who received IO or 2I would have blood type O. Here is the checkerboard. Since 3 alleles for ABO blood group are found in a population, it is an example of multiple allelism. This question carries 5 marks, and this answer is quite long, but it really helps you. The next question is, what is the causative agent of monkeypox and how can it be transmitted to human? So here is the answer. Monkeypox is a viral disease caused by the monkeypox virus MPXV. This is a short term, a member of the orthopox virus genus. It is closely related to variola virus, which causes smallpox. Monkeypox can be transmitted to humans primarily through direct contact with infected animals. The virus can spread from animals to humans through several routes. Number 1 is direct contact with infected animals. This can occur through handling or infected animals such as monkeys, squirrels, or rodents, or by coming into contact with their body fluids, including blood, respiratory secretions, or lesions. And number 2 is consumption of infected animals. Eating undercooked meat from infected animals, especially rodents, can lead to transmission. And the last one is human to human transmission. Monkeypox can be transmitted from person to person through respiratory droplets or contact with respiratory secretions, skin lesions, or body fluids of an infected individual. This mode of transmission is less frequent than direct contact with animals, but can occur during close contact, such as caring for a sick person or through contact with contaminated bedding or clothing. This question also carries 5 marks. Question number 28. The question is, a taxonomist has found 3 related species of an insect with 3 different variations of wing pattern. He has coded the variations as 0, 1 and 2 to establish taxonomic relationship among the insect species. What is the basis of taxonomy that he is following? Enumerate 2 sources of variation in a population. This is the answer. The taxonomist is using a numerical coding system 0, 1 and 2 to establish taxonomic relationships among the insect species based on their wing pattern variations. This indicates that the taxonomist is following a numerical ordinal system for taxonomy. 2 sources of variation in a population are genetic variation. This refers to a diversity of genes within a population. Genetic variation arises from mutations, recombination during sexual reproduction, and genetic drift. It can lead to differences in physical traits, such as wing patterns in case of the insect species, as well as differences in behavior, physiology, and other characteristics. Next source is environmental variation. This refers to variations in the environment that can influence the phenotype of individuals within a population. Environmental factors such as temperature, humidity, availability of resources, and exposure to certain substances or conditions can cause variations in traits. Thus, these 2 sources of variations play a crucial role in evolution and the formation of distinct species. If you want these answers in Bengali, please comment me in the comment section, and also hit that notification bell so you never miss a video. Are you learning happily? Are you make out these answers? If you get the point, then give me a thumbs up and share with your friends. So the next question is, what is happy hypoxia? How can it be readily detected? Happy hypoxia, also known as silent hypoxia, is a condition in which an individual's oxygen levels in the blood are dangerously low, hypoxemia, but they do not exhibit the typical signs and symptoms associated with oxygen levels, such as shortness of breath and respiratory distress. It is called happy hypoxia because patients may appear relatively comfortable and alert despite the severity of their condition. Detecting happy hypoxia can be challenging because the usual indicators of hypoxemia, such as shortness of breath, cyanosis, bluish discoloration of the skin, and increased respiratory rate, may be absent or minimal. Thus, it is also known as silent killer. The answer of the next part is happy hypoxemia detection. First one is pulse oximetry, arterial blood gas, short form is ABG, analysis, and clinical assessment about respiratory illness, heart rate, and mental status to evaluate the severity of the condition. Happy hypoxemia is most commonly associated with respiratory syndrome, including COVID-19. Question number 30. Suppose you have been asked to carry out an experiment wherein you have been given a test tube containing an enzyme that binds to substrate, and that enzyme has a fluorophore attached to the substrate binding site. While doing such study, you have added a urea solution in the test tube. How will you follow the effect of urea addition, and what would be your possible observation? These questions are from JBNSTS sample paper, and this question is divided into 2 plus 3. Answer. There are several ways I can follow the effect and make observations. Here's a possible approach. Number 1. Baseline measurement. Before adding urea, I would measure the fluorescence intensity of the enzyme substrate complex in the test tube using a fluorometer or a similar instrument. This measurement will serve as the baseline or control. The next one is urea addition. I would add a known concentration of urea solution to the test tube containing the enzyme substrate complex. The concentration of urea can vary depending on the specific experimental design and objectives. My next approach, incubation. After adding urea, I would incubate the test tube at the specific temperature and duration to allow the urea to interact with the enzyme and substrate. Next and last, fluorescence measurement. After the incubation period, I would again measure the fluorescence intensity using the same fluorometer. This measurement will indicate any changes in the fluorescence due to the addition of urea. Coming to this point, possible observations. If the fluorescence intensity shows any minor fluctuations or remains unchanged, it suggests that urea does not significantly affect the binding of the enzyme to its substrate. Number 2 observation is a decrease in fluorescence intensity compared to the baseline measurement suggests that urea may have disrupted the binding of the enzyme to its substrate. Urea is a denaturant that can unfold proteins and disrupt their active sites, leading to reduced binding affinity and enzymatic activity. And the last observation is an increase in fluorescence intensity. Urea can induce conformational changes in proteins. It suggests that urea has altered the microenvironment around the fluorophore, leading to enhanced fluorescence intensity. These are the observations